All right, so hi everyone. Today we're going to talk about the Mendelian randomization identifies causal genes in non-small cell lung cancer. And it's presented by me, Adam, by Terry, and by Liao. And to begin with, Terry's going to give us a brief introduction of the background.
Sure. These days, non-small cell lung cancers accounts for around 85% of lung cancers. It's still leading a cause of cancer mortality. And however, the large-scale data sets like GWAS found many associated SNPs or genes to these lung cancers, and also, but many of them are in non-coding regions. So we know the loci and the associations, but we still do not know which genes causally drive the non-small cell lung cancers. And this is where Mendelian randomization becomes powerful. The concept of Mendelian randomization builds on the concept of instrumental variable. Sometimes we want to show causal effect from exposure to outcome, but the exposure is confounded or not experimentally controllable. In our case, the gene expression as an exposure might be affected by various confounding factors like nutrition, so we cannot determine whether the gene expression directly has a causal effect to our outcome, which are diseases. So to deal with this problem, we create an instrumental variable z to estimate the effect of exposures A on outcome Y. And conceptually, this is using a z-driven variation on A to identify the A to Y causality. And there are some assumptions that need to be made for this instrumental variable. For example, it changes the treatment or exposure without being affected by confounders. So it's not like the U is not pointing towards Z. And it affects outcome only through the exposure, not directly. So there's no direct arrow from Z to Y in this stellar graph. And this assumption allows us to extract the causal part of exposure to outcome relationship. In our case, we use single nucleotide polymorphisms, SNPs, as our instrumental variable. Many disease-associated SNPs are non-coding and regulatory, and they influence gene expression rather than the protein structures. And in our case, we focus on the cis-EQTLs, which are SNPs near the gene, which increases the chance we capture direct regulatory effects. By using the gene expression and exposure in SNPs as an instrumental variable, we get a biologically plausible and testable causal pathway from SNP to gene expression to cellular mechanism to a non-small cell lung cancer risk. And in our research, we do two-sample Mendelian randomization, which means the exposures and outcomes come from different data sets. We get exposure data from EQTL, and we get the outcome data from GWAS. For this to work, there are like a few other assumptions, like details, we need to figure out. And in our setup, we use SNPs as our instrument, or use gene expression as exposure, and use a non-small cell lung cancer risk as the outcome. Uh, and there are three assumptions for Mendelian randomization to work. The first one, relevance, means that the SNPs should need to be strongly influencing the gene expression. And in our case, we select the significant SNPs among the CIS-EQTLs with f-statistics greater than 10. And the second assumption, independence, means that the SNP is independent of confounders. This is justified by Mendel's laws because confounding factors like nutrition is not supposed to affect your SNPs, which is part of your genotype, which part of your gene. And the last assumption of exclusion restriction says that SNP influences the non-small cell lung cancer only through the expression of the gene. And there are no other exporter factors, so there's no horizontal pleiotropy. And we verify that through various methods in our research. Some prior works look at the metabolites, immune trace, biomarkers, white blood cell trace, etc. No study has systematically tested all available genes as causal drivers for non-small cell lung cancer. We use all available CIS-EQTL genes from EQTL gen, GTEX, and so this might be the first comprehensive gene level Mendelian randomization for non-small cell lung cancer. We test each gene's expression as an exposure for Mendelian randomization, and our goal is to produce a ranked list of causal gene candidates.
So basically, I will walk you through the methods part. So first of all, I'll introduce how we calculate the causal effect in this Mendelian randomization study. So basically, it depends on different numbers of SNPs. When we are only handling with like one SNP or one instrument variable, we will be simply using the wall ratio estimator. That is, we'll be using the estimated effect of SNP on the outcome divided by the estimated effect of SNP on the exposure. While we are handling with like multiple SNPs, basically, we'll use the inverse variance weighted method. That is based on the wall ratio we derived in before, and we add a weight on it. So intuitively, we would consider SNP with smaller variance will receive more weight. And after these calculations, the estimated causal effect will be stored in the format of odds ratio. So we'll also be looping over each candidate gene to do the harmonization and stagger test. Basically, when combining exposure and outcome data from different GWAS, the same SNP might be coded differently. So we introduce harmonization to flip the estimates, so both refer to the same allele. And secondly, we will be handled forward and reverse strand differences. Third, we drop SNPs whose allele frequencies are ambiguous. And last but not least, we'll remove SNPs that can't be reliably aligned. And we also perform staggered directionality tests to see how well SNP explains gene expression and how well SNP explains. So an important factor we'll be comparing is R squares, which is the proportion of variance explained. And based on one of our assumptions, which is exclusion restriction, we would be expecting the SNP should be strongly associated with the gene expression, while at the same time weakly associated with the outcome. And we'll be using Fisher's Z transformation to compare two correlation coefficients. We will also do sensitivity analysis and basically test on two things. The first one is pleiotropy. The second one is direct effects. And basically, we do this when there's more than one SNP. And first of all, we'll do cultural Q tests on heterogeneity. Basically, what these tests do is checking do all SNPs given consistent causal estimates. And as I mentioned before, each SNP gives a wall ratio. And if all SNPs are valid instruments, we will be expecting these ratios to be highly similar. And this could also be reflected by the test statistic. And if the Q value is low while the P value is high, then that means like SNPs agree with our assumptions. While if it goes the other way, well, which is like high Q value and low P value, that says SNPs disagree with our assumptions and probably suggesting horizontal pleiotropy, invalid instruments, or probably different probability structures. We'll be also doing the MR-eager intercept. Basically, what this do is check do SNPs have direct effects on the outcome. And what we'll do is we will perform a regression of like beta outcome on the beta exposure and checking two things. The first one is intercept. The second one is the test statistic. And if the numerical value of intercept is 0, that says there's no directional pleiotropy. While if it's not 0, that stands for like there's average pleiotropic effect across SNPs. And we'll be also checking the p-value, and our null assumption is like there's no evidence of directional pleiotropy. And if the p-value is larger than 0.05, we'll be failed to reject the null hypothesis. Well, if the p-value is significant, then we will reject the null hypothesis and adjust our conclusion accordingly. We'll be also doing the one-out analysis, and that is the result driven by a single SNP, and we will rerun the inverse variance weighted analysis n times. Here, n stands for the number of SNPs here, and each time we remove one SNP, and we will be then compare the results. And if one SNP changes the result dramatically, that says that SNP is influential or problematic. We'll be also talking about like a new way of measuring the causal effects. The methods I introduced basically testing each gene independently. So, however, the genes operate in the pathways, and co-expressed genes often have coordinated effects, while weak instruments may miss truly causal genes. In addition, the enrichment test is purely descriptive. Hence, we try to incorporate the analysis of enrichment pathway into the MR framework. That says genes connected in the network, they share the information, while the weak genes borrow strength from the neighbors. And we hold the belief that connected genes should have similar causal effects and then adjust the causal effect estimator. And here, W is weight of each gene, while the parts on the others after the adding sign would be the would be like the adjustment after you incorporate the network. Here the lambda stands for smoothing parameter, A stands for adjacency matrix, while the D stands for lambda times D stands for total borrowed weight.
All right, so I'm gonna talk about the preliminary result, which is the full result of the standard Mendelian randomization here. So to begin with, we have this volcano plot as the most direct and simple, straightforward visualization of the result. So this type of volcano plot is a type of graph, the most important graph from nearly all Mendelian randomization studies. So on the x-axis, we have the odds ratio, as Leo mentioned before. Basically, an odds ratio of less than 1 indicates that the gene or whatever exposure you are using has a protective effect against the disease phenotype. When the odds ratio is larger than 1, we have a causal effect. And then additionally, we color the source based on we color the point based on the source of the of our exposure. So for instance, sorry, we'll shape them. So for instance, the circle ones are from blood and the triangle ones are from the lung. And then the horizontal asymptote basically just simplifies the statistical significance threshold, which is FDR less than 0.05. And additionally, we have 9 significant results in total. When stratifying that by tissue, we have 3 significant results from blood and 6 significant results from lung. And when stratifying that by direction, we have 4 protective effects and 5 causal effects. And here I'm going to dive a little bit deeper into the specific results we have through this first plot. And basically, the first plot has the interval, the 95% confidence interval of the effect range of each gene. And then you also see the method the result is derived from, as Leo mentioned before. And we also have the significance value here and also the source of them. And basically, the number of SMP is also listed here. So we categorize all the results into 3 categories. The first one is that we have concordance results from similar to previous studies, and the second one is that we have contradictory results to previous studies, and then the last one is that our results provides further elucidation of previous studies. So basically, the concordance results are pretty straightforward. We can see that the HYKK is basically protective in our result. That is demonstrated by Wang et al. In 2025. And in contrast, the link 00173 and then the PSMA4, those are both causal and are proved from previous studies to non-small cell lung cancer or other types of cancer. Besides 2 notable cases we have here diverge from previous experimental findings. For WASIR2, found out in 2024 shows that it has oncogenic functions using cell line assays. However, our Mendelian randomization here shows that it is protective. Additionally, the NPAS4 in our results, it is more causal to the non-small cell lung cancer, whereas it is more protective using the Drosophila and mammalian cell line model in previous studies. I just want to note that these divergences may be driven by different factors. For instance, previous studies were not based on the strict randomized clinical trials that Mendelian randomization tries to mimic. And additionally, their models like cell line models or Drosophila model cannot capture the whole human status that Mendelian randomization tries to capture. On the other hand, Mendelian randomization also has drawbacks and need to be further validated through both experimental and statistical approaches. And then lastly, we have a category of our result providing further elucidation to previous studies. For instance, for IREB2, basically for that gene, previous studies only said it's a biomarker related to the prognosis and correlate with autophagy related ferroptosis and immune infiltration in renal clear cell carcinoma. But now we see a direction of protective effect in non-small cell lung cancer. Additionally, for the MMP3, the literature is mixed, with some clinical studies suggesting increased risk and some showing no association. Our Mendelian randomization shows that it supports a protective effect. Lastly, the small RNA SNORD35 has been proposed as a diagnostic biomarker in previous findings. However, no functional relevance was mentioned in previous studies, and now we see that it is a gene that can be causal to non-small cell lung cancer. And this is a leave-one out analysis, as Leo mentioned before. Basically, leave-one out analysis just try to see when there are multiple SNPs contributing to a gene and thus contributing to a disease. We just want to see whether all those SNPs give a consistent effect across. Yeah, and basically, the only two genes that has multiple SNPs are the PSM4A and the IREB2 here, as shown by this first plot. So PSMA4 has 3 and IREB2 has 5 SNPs. Yeah, and on the graph on the left, although the first SNP seems to deviate from other SNPs in PSM4A, it's not statistically significant. However, we see that the third SNP in IREB2 is more significant and it is statistically significant. Basically, that means that when you leave that SNP out, the causal effect of IREB2 will be very different from including that SNP. This does not necessarily mean that the Mendelian randomization for this gene is not reliable. It just means that we have to be more cautious and potentially we need to establish more concrete evidence through either experimental approaches or other statistical analysis. So this just gives us a heads up. And finally, we have a supplementary analysis that is not included in our original proposal. So basically, after finding all those genes, we try to do a pathway enrichment for those. Basically, the logic is overrepresentation in gene ontology process. So basically, we try to see whether the chance of those significant genes appearing in these certain pathways is statistically significant compared to random chance of those genes disappearing anywhere in the entire gene list. And basically, a significant result indicates that the appearance of these genes means certain pathways are potentially implicated. For instance, in the first one, in cellular amino acid metabolic process, it basically is just saying that because of the presence of our significant genes, a biological system is probably going through this process. And we do not observe any statistical significance at the p-value of 0.05, so we decided to be more lenient and to use a p-value of 0.1. So basically, we do see a bunch of pathways, but it's harder to provide further distinguished or classification of those pathways. So we just basically made a graph of the top 10 enriched pathways. And yeah, those are all statistically significant at the p-value of 0.1. And this is only a supplementary analysis. And then this is our timeline. So basically, from the progress to date, we have completed the standard Mendelian randomization and getting the 9 significant results that I just talked about. And we also did the complementary sensitivity analysis to ensure that our result is concrete and valid, and also a pathway enrichment analysis as a complementary analysis. And also at the same time, we have been trying to build this new model to adjust the Mendelian randomization MR predicted causal effect based on the assumption that a lot of times multiple genes are co-expressed. So basically, through this new model, we just want to see whether our new model will get some different results from the standard model, based on the assumption that a lot of genes appear together. Whereas in standard model, there's no this assumption. And basically, in the next week, we try to complete the new model. We try to turn the statistical concepts into actual codes. And then in the next week, we will try to compare the new model to the standard result. And it will be a really challenging process, but we think that this work will be really beneficial. And even just for the standard Mendelian randomization itself, we already believe that the result is significant because we don't think any studies previously have done exactly the same thing before. And yeah, this is it. This is our references. And thanks so much for listening. And please reach out if you have any questions.